I would like to introduce the next award. This is the Lennart Philipson Award, which was initiated and launched by EMBL in 2014, thanks to the initiative of Gabor Lam and Jürgen Bauer. Both of them are here today with us. They keep supporting this fantastic award, and let's give them a big round of applause. Thank you so much. There has been until now 12 winners of this prestigious award, and the prestige is also measured by the fact that one of them is a Nobel Prize winner. We don't have yet the video, but we are working on it. So next year there will be a video. The recipient of the Lennart Philipson Award for 2026 will be introduced by Elena Seiradake, to whom I would like to pass the microphone now.
Fantastic. Hello, everyone. This is such a huge honor to be here, to be introducing Stephen. I'm absolutely thrilled. And although I've been a non-dormant alumna for about 20 years, I find that every time I come here, I walk those steps up to the ISG hotel. I find those, it brings back so many memories. And then coming here to EMBL is just absolutely fantastic. So my brief for today, very important, wear your best dress, done, sparkling. Second, introduce yourself and Stephen, but stick to 7 minutes. Now that is challenging. I mean, myself, fine, but Stephen, in 7 minutes? And then, also Stephen doesn't like to be made a fuss about, so I'm glad he's sitting really far away, so I won't see his eyes rolling quite so much. So let's get started with me. Okay, I'm a professor of molecular biology at Oxford University. My lab is really interested in how cells use cell surface receptors and their ligands to interact, and that provides communication between cells, and that's really, really important in tissues. We've got to focus on brain development, on cancer, and on autoimmune disease. And what I find most fascinating about our work is how, by looking at how the same receptors or similar ones function in very different systems, you can start to get at that fundamental logic of how cells communicate. And that's what I love about biology, how it's both super complex and yet simple. So, but my whole scientific journey actually started in Stephen's lab, where I was a pre-doc in that fantastic course that's still going on today, the pre-doc course. Starting in 2003. And Stephen was an amazing, amazing supervisor. Now, I tried to think about why he was so good. Actually, a lot of us from his lab did really well afterwards. And I think there are two key ingredients to his success with being a great mentor. One is obviously his intellect, like he's got an amazing scientific brain, and it didn't take us very long to know that we could absolutely trust his judgment and advice. But the second one are his feet. Now, Stephen has a very magical way of just appearing out of thin air, completely silently. So he'll just appear next to you, and he'll use that, that element of surprise to get you to tell him what you're actually up to. So he'll go like, "So, what are you doing?" And you go, "Oh." And then that was brilliant because we couldn't sugarcoat, and try and quickly hide all the not so good things we were doing, but we would tell him what we were doing, and he always knew what we were up to and gave us really good advice. So taken together, that was really fantastic. In fact, he was so good with this skill that I often wondered that the MI6 didn't try to poach him. We all tried to be little Bonds, like back at that day. That was a lab, and I was there too. But now really properly to Stephen, not about my memories. So Stephen, I think, was born in London, but then he grew up in Norwich, which is where his parents moved to. And that's also where he met Alison, his future wife, who is here in the audience today. And they do say that next to many great men, there's a great woman. So there were many twists and turns. I'm not going to try and explain how they managed to both end up in Grenoble, but they did manage so. They're both scientists and had a wonderful family. So you can see on the picture here, their sons Jeremy and Martin, and also Jeremy's wife, Rocio. I think this is a very recent picture. And both Jeremy and Martin are also scientists, which I think Stephen is very proud about. And this just shows you how inspiration can work. So, because I was trying to read about Stephen's interviews, and I noticed that he often mentioned his parents, who were also scientists, so they clearly inspired him as well. His father was a physicist, a professor, and his mother was a biology teacher. And I think Stephen's research really lies at that interface. He is comfortable in both of those areas. And he did start as a physicist by reading physics at Cambridge and then doing a PhD at the Imperial in theoretical physics. But then when he came to Grenoble, that's when he started working on virus structures with Bernard Crowe. And I think that's key because that's when he met biology and he realized that he can bring a lot to that field. But he, I think, wasn't entirely satisfied with the method he was using initially, which was neutron scattering. So he went to Don Wiley to train in X-ray crystallography, and that has really shaped a big chunk of his career. I think what I want to mention is, well, here you can see Stephen, a young director, I think, at the EMBL in Grenoble. But what he really managed to do, he managed to lobby, to push for X-ray crystallography to become important in Grenoble. So the first big synchrotron, third generation synchrotron in the world was built in Grenoble, and that's also thanks to Stephen. He's also pushed a lot for the method to be integrated. So X-ray crystallography to be integrated with other structural methods. So he was leading or co-leading initiatives such as Instruct and the PSB, which has, I think, been very, very powerful, not just in Grenoble, but across Europe. So in brief, so I think many of his great discoveries had to do with X-ray crystallography initially, although obviously, like many of us, he's doing cryo-EM now. I'm not going to say very much about the science. I don't want to run out of time. So all the amazing stuff about influenza polymerase and Rick, I hope Stephen will explain to you. I just wanted to very cheekily take this opportunity to show you unpublished data, because you can't come to EMBL and not show unpublished data. So I still have unpublished data from the time in Stephen's lab, and that was on the 20 years later. And that was from contributing to that amazing project on tRNA synthetase, which obviously led to a marketed drug, Caridin, and there are other drugs in the pipeline. But here you can see it, world premiere, the mitochondrial lucile tRNA synthetase editing domain. Ta-da! So this is a protein which, so we have a cytosolic and a mitochondrial version of this protein, and the cytosolic one very much responds to these drugs, and there's the whole mechanism behind it where they block the editing site of that domain. But the mitochondrial one is more related to bacterial proteins, and it's different, and it has actually lost its editing function, and it cannot bind to these drugs. So it is actually quite cool, and we should still publish this, Stephen, I think. I want to end now and just say I'm really glad to be here and to be celebrating with you. Many of your friends are here to celebrate with you, Stephen. I will put just a representative picture on. I don't have one with all of your friends, so I'll just put this one up because you're both smiling so nicely. So I would like to invite Stephen to come and get his prize now.
Okay, so this is one of those days where you're a hero, but just for one day, fortunately. Is it ready? No. Well, so I should start by saying I'm very, very thankful and honored to receive this award. It's connected to Lennart Philipson, who I greatly admired, and I'll say a bit about that in a minute. And it's also connected because of it's sponsored by Emblem, and I'm also indebted to Emblem for most of what I'm going to talk to you about, the fact that it could happen. And so what I'm going to talk about is my forays, as I call it, into translational research from always based on fundamental research. And I'm going to give 3 brief forays, which are each going to start with an image, a photograph. And the first is passion flowers. So here we go. These are beautiful flowers. And more on the botanical level, though, they have an interesting feature, which is that they are anisomerous. Now, I don't know if anyone knows what that means, but it means that the petals and stamens are pentamers, and the carpels are trimers. Can you see that? Yeah. Right. So what's that got to do with anything? Well, it's got to do with adenovirus, because the penton of adenovirus is also anisomerous in exactly the same way. This red protein, which is actually made up of 2 different proteins, is called the penton, and it comprises the penton base, which has got 5-fold symmetry, and the long fiber, which has 3-fold symmetry. So it's got this interesting mismatch. So the primary adhesion is done by the fiber, the long fiber, which binds to a receptor called the Coxsackie and adenovirus receptor, whereas the penton base, which is integrated in the capsid, binds to integrins, and that's responsible for internalization. So early on, I studied this. This is the first project I studied after coming back from Harvard. And we eventually determined structures of this very long fiber molecule, the trimer, and the pentamer, which looks like another flower. It looks like a tulip. So, and then in the same work that we did on the pentamer, we were able to solve this symmetry mismatch problem in that we could soak in peptides which corresponded to the N-terminal of the fiber protein, and we found they bound in the penton base in this way, which mimics the passion flower. So, what's all that got to do with anything? It's because the penton is also the special red spot in the EMBL logo. You can all see that. And of course, that links to Lennart Philipson, who invented, as we all like to think, that logo. And he indeed related it to adenovirus. He regarded the spots as the hexons representing the labs in Europe, and EMBL is the red spot. So EMBL is this red spot which is anisomerous. And Lennart and I shared this interest in adenovirus, and he, in fact, very much earlier had identified this, the fact that adenoviruses and Coxsackie viruses had the same receptor. That was back in 1976. It wasn't until 20 years later that he and others identified the actual receptor, which is this, what's now called the adenovirus and Coxsackie receptor, which is a tight junction protein. And then we worked on this host cell interaction and studied the fiber-CAR interactions. And Elena, in fact, did her PhD on some of this. And why am I saying this? Because this was the first foray into translational research. Our work eventually led to a collaboration with Transgene in Strasbourg, because at the time, the adenovirus was the key candidate as a gene therapy vector. And the whole major goal, as it says in this paper we co-published, was to be able to target adenovirus to any organ or cell type that you wished. So understanding these interactions was critical. But that actually didn't go very far, even though we had a joint patent, because soon after there were lethal results of an adenovirus clinical trial, which held things up. But as you know, it's come back as a vector for cancer therapy and of course, in vaccines. So moving on to the second foray. This starts with this picture, and it takes two to tango. And this was actually a song written in 1952 when I was born. And I first thought about using this analogy when we had a meeting in Argentina, which probably Ian remembers, when Argentina joined as an associate member. And so what's the thing here? Well, the thing is, it's this tRNA synthetase complex. Lucile tRNA synthetase is one of the 20 synthetases that specifically charges a specific tRNA leucine with leucine, which then is used in protein synthesis on the ribosome to incorporate leucine in protein chains. So we studied this an awful lot in the 90s and 2000s. And the interesting thing was that these two molecules have a dynamic, they have a dance. And the end of the tRNA is first in the amino acylation site to charge the tRNA, and that's part of the tango dance. And then the end of the tRNA flips into the so-called editing site to be proofread to make sure it's the right amino acid, and that's another part of this dynamics. And we studied in great detail at that time what were the specificities of each of these two active sites that one could majoritively recognize leucine, but occasionally make mistakes, and the other site which would exclude leucine and only remove the mistakes from the end of the tRNA. Yeah, so there. Now, where do we go from here? The next part of the story is this man, Dickon Ali, who was head of discovery biology at Anacor. And Anacor specialized in boron chemistry, and they had a proprietary library of boron compounds. They screened pathogenic fungi and came up with a broad-spectrum antifungal, AN2690. And so we met at this conference in Bangalore. He told us this information and the critical fact that resistant mutants to this compound all occur in the editing domain of yeast lucile tRNA synthetase. So this compound apparently targeted the editing domain of lucile tRNA synthetase, of which we were the experts. So we collaborated, and within a few months, we had a structure of this tRNA where the dynamics is killed because this compound makes a covalent adduct with the end of the tRNA, and it blocks the whole tRNA into the editing site, and all the beautiful dynamics of the amino acylation cycle stop, and you killed protein synthesis, basically. So this was the mechanism of the drug which we very quickly established in this early work. And this led on to this same compound, the original compound, AN2690, eventually being approved as a treatment for toenail fungus, which some of you may have and some of you may not. But more probably do than we'll tell you. So this drug is now available and makes around 170 million a year, but we had no IP on that compound. Okay, that's all very well, but let's be serious. What about tackling the antibiotic resistance crisis? So this is the most important slide in my whole talk because it is the key to a whole series of other drugs based entirely on the structure, where if you notice in orange is the true substrate of this editing site, which is a non-cognate amino acid other than leucine. And in purple is the compound. And you see that the true substrate makes these 3 extra hydrogen bonds, which the compound doesn't. And so we put them in. We created a new compound with this extra amino methyl group that the Anacor chemists made this. And this, we verified, did what it was supposed to do structurally. And this compound, plus another substitution, which is in the bottom, which was also designed using the crystal structure, which improves the interaction with the tRNA. This led to a very potent broad-spectrum gram-negative antibacterial called AN3365, but now it's called epetraborole. So this was very exciting because this hit all the drug-resistant nasty bacteria that you can think of. So it went into clinical trials as a new mechanism, gram-negative antibacterial. But in the 2 phase 2 trials run by GSK against complicated urinary or intra-abdominal infections, which are really tough issues, there was a problem of emergence of resistance. And so these were eventually stopped. And GSK abandoned the compound. This is typical of what happens. Most compounds fail. But didn't stop Anacor being acquired by Pfizer for $5.2 billion. Not because of our compound, but for a new boron-containing compound, which is now available as called crisaborole. It's now on sale as a atopic dermatitis drug. But anyway, the previous compound is still in clinical trials for targeting different diseases by AN2 Therapeutics, which was the Phoenix company of Anacor after they were taken over by Pfizer. But let's move on, because at the same time we tried out all kinds of things, and we discovered with Anacor that and GSK that if you put a chlorine atom here, you gain very high specificity for tuberculosis lucile tRNA synthetase, and lose the affinity for E. coli and other bacteria. And this compound is faring much better. This is another totally new mechanism anti-TB drug. It's now called ganforborole, and it's still in clinical trials. Some were published recently. And this is based on the work we did 12 years ago now. So it takes a hell of a long time to get anywhere with these things. Okay, I better speed up because I have 1 minute left. So we went through tavaborole, etraborole, and ganforborole, and you've heard that the last one is still active, and let's hope it works out. And these are some of the people involved in all that work. And it was a great time at the time. So the third final story starts in Singapore. Some of you may recognize where this is. And I was there in 2019 for an influenza meeting. And at this meeting, with grand pomp, Roche announced a new anti-influenza drug, which was called Xofluza. That was the trade name, and the other name is baloxavir. And they had this slogan, "Tell the flu that its time is up." In all great seriousness, they thought they'd solved the flu problem. Well, you have to sell it somehow, right? So where did all this come from? Well, this is targeting the influenza polymerase, which uses these 2 domains, the cap binding domain and the endonuclease domain, to cap snatch transcripts from Pol2 to generate capped primers to do transcription of viral mRNA. So I don't really have time to go into all that. But just focusing on the cap binding and endonuclease domain, so you get this cleavage, and then the cap primer is rotated to prime transcription of viral messenger RNA from the viral RNA genome. So early on, we solved the structures of these two domains as individual domains, long before we got a structure of the whole polymerase. And these, we worked with Emblem to patent just the structures. And we founded this company, Severa, in 2009. And Oliver was the chief executive officer. Both of us here are in white coats, but we never did any lab work, of course. And we got an EU project. But things really took off when Oliver negotiated a contract, a partnership with Roche. And there we worked with Klaus, who was head of virology at Nutley in the USA, who, by the way, is an alumnus PhD from Grenoble, head of virology. And we had a fantastic period working together. We solved lots of structures of compounds made by Roche and Severa chemists, in both domains. And we had 3 X-ray structure patents. We had 15 composition of matter patents. But then what went wrong? Because we don't have a drug in our name. So what happened was that in 2013, Roche took over what they called sole responsibility, which basically kicked us out, and because I think we were just too slow, they could do things more efficiently themselves. And they actually worked incredibly hard and did lots of stuff. But in the meantime, 2 competitors, Vertex and Shionogi, came up with pimovadivir and baloxavir, which were much better than anything we came up with. And so what did Roche do? They switched camps, and they abandoned us and made a partnership with Shionogi. The Vertex one failed in phase 3. You know, imagine the amount of money that went into that before it failed. But this one, they gave us a bit of money, a token amount of money. On whose scale? On whose scale? So baloxavir got approved in 2018 and 19. And irony of ironies, Shionogi then came to me to collaborate on the structural basis of the mechanism. So this tells you what translational forays end up in. And then that wasn't that. So that's the background to this launch of this new drug, which is quite an amazing drug. It's now approved in many places in the world, but it's not yet available in the UK for technical reasons. Peak sales are more than 500 million a year, or that fluctuates. Again, but there's a but, because this compound, this is the work we did with Shionogi, binds picomolar affinity in the endonuclease. But even in the clinical trials, they had resistance emergence in the patients. And we characterized this I38T mutation, which is just one methyl group, right? And but it lowers the affinity for the drug. And, a few months after the launch of the drug, people were saying, miracle drug foiled by resistant strains. Okay, so thing is, Xofluza-resistant viruses are not generally spread because, as we showed in this paper, they are less fit, so they don't compete with wild type except in the presence of the drug. And in fact, it's not apparently a problem in the treatment, but anyway. But then look at this. Global Xofluza market estimated to reach 4 billion by 2034, driven by acceptance all over the world and government pandemic stockpiling. So, we missed out a bit, just a little bit, in not developing our own drug. So here's the last slide with some lessons. So forays into translational research can be fun, rewarding, but incredibly frustrating. I think that sums up just drug development. The compound can be great in vitro, but useless in vivo. Most drug candidates fail. But this is what's interesting. Pathogens resist and are very good at resisting. But sometimes you can get away with that, like with the Xofluza drug. It all depends on whether the drug resistance get established in the wild type or the spreading pathogens. The other thing is big pharma can have totally their own agenda and can drop you. They just care about dollars. And it's a shame we didn't have royalties from a $1 billion drug. We were not far off, you know. Wouldn't you have liked that? Right, so finally, thank you to everybody. I have too many people to thank to do it in name by name, but some really important people were Dick and Ali, Oliver, Klaus, Shinya Omoto, the chemical biology core facility people who gave us some tremendous support, EMBL, ESRF infrastructures, Emblem, and Jorgen, I guess the guy I worked mostly with. Is he here? Where is he? Yeah, I mean, we had 15, 20 years of collaboration really closely. Thank you. And of course, Lennart Philipson for his confidence back in 1989. Thank you.